very much. Thank you for the nice introduction, and thank you for inviting me here to speak. I've never actually been here, and so it's great to finally see this great center. Although the Boston roads on the way here weren't ideal. So I'm going to talk today about the challenge, the promise and challenge of voxel-based measures of intrinsic connectivity. And for those of you that do a lot of fMRI, some of the beginning parts here will be pretty straightforward for you, but I figured it would be a mixed audience, so I have a wide spectrum of things to talk about here. I'm going to talk about what I now call classic fMRI, which is a task-based fMRI studies, and then we'll move into conventional resting state functional connectivity, and we'll look at measures of intrinsic connectivity or voxel-based sort of measures of intrinsic connectivity. And I know a number of people here have already published on some of this stuff, and so I'll actually refer to some of those papers. And then I'll talk a little bit about applications of these techniques clinically. So in classic fMRI, as you know, the most common experiment is a block design experiment where we might have a run that takes, say, 4 to 5 minutes to do. And each block may last 20 to 40 seconds. And we acquire images continuously throughout the run, and we separate those into activation and control. And we're basically collecting these brain volumes as we go. We can then sort those, do some motion correction, sort them into the different conditions, and do some statistics on them to ultimately get activation maps. And this is great for things like surgical planning, where you might want to look at motor cortex function, or it's great for cognitive neuroscience, where you might want to work out what specific areas are involved in specific aspects of tasks. Here we have some graded changes in the motor organization as a function of pathology. And you can see here, for example, the blue motor cortex, which should be on the left, has migrated over here to the right. And so it's a great technique for actually looking at this sort of plasticity of the brain as well. And just to summarize what goes into an activation map, we have these, say, T1 flash or anatomic images, the functional images that are sensitive to the blood oxygenation, a statistical parametric map, which we calculate comparing the red and the blue read task conditions, and ultimately we threshold this at some arbitrary threshold, and we overlay it onto this map here. And so that's how we get these maps that you are very used to seeing probably. So classic fMRI, even though it I'm going to talk about its limitations. I don't want to say nobody should do classic fMRI. It's very useful, as I said before, for cognitive neuroscience studies and for the types of mapping studies for surgical planning that I just showed in the motor cortex there. One of the problems is that in survey studies, the limitation on the number of tasks is somewhat prohibitive. You can only do maybe 3 or 4 tasks. We always argue with our neuroscientists as to how many tasks they can do with valid statistics. And 3 to 4 seems to be a good number in the hour-and-a-half time that is typical for a study. And this is potentially a major limitation for characterizing the impact of disease on the brain's functional organization. And also for looking at the impact of drugs on brain function. And in terms of drugs, I mean either the effects of drugs, so drug addiction studies, for example, or in drug development work where drug manufacturers trying to see what the impact on the CNS system that a drug may have that they're developing. So furthermore, people are very interested in brain function and genetics, and to characterize certain gene expressions can be very difficult with only a few tasks. Primary concern is what if those 3 or 4 tasks that you run are not the key tasks for separating the genetic expression or something like that. So connectivity may have a role here in the sense that it is more general, and you can look at the whole cortex. You're not just limited to a few areas of the brain that light up. You can actually look at the whole brain and the subcortical function as well. It's task-free, we could say, or it's at least less task-dependent. There's 2 types of connectivity, as you well know. Some of the leading experts in DTI and anatomic connectivity are here, and I've already seen some early results from Van Wadeen in the Connectome study, and it was pretty impressive what's coming out of here. So we have anatomic connectivity in DTI and also functional connectivity. And I'll just briefly review the difference between these. So in anatomic connectivity, we can get these fiber maps that show the pathways, and we can isolate certain fiber bundles and things like that. We can also look at fractional anisotropy and look at the directional specificity of the diffusion in the brain. This is usually a white matter measure. And we're exploiting the structure of the white matter in order to look at these tracks. So this is great, and it can be used in evaluating disease and evaluating, certainly with genetic phenotyping studies moving forward, people will be looking at this. And in terms of isolating specific bundles, fiber bundles like the uncinate fasciculus here, and then looking in particular at characteristics of those bundles, it's an excellent technique for that. In general, DTI then provides a whole brain assessment of the microstructure of the white matter. It can be very important in developmental disorders, particularly when there's neuronal migration problems, for example. And it can also be a very sensitive indicator of degeneration. But it doesn't really provide any information on function. I know as part of the connectome, one of the goals is to try to relate some of the underlying structure with the underlying function. And so hopefully some progress will be made in that area, but right now there isn't a good understanding of what the link between the two is. Fiber tracking itself can be very difficult, and ultimately every region of the brain is connected to every other region, so we know we can get from A to B through the fibers somehow. Functional connectivity, on the other hand, allows examination on all brain circuits, and now we're talking about the gray matter and the cortex, without dependence on a specific task. So we're not just looking at single activated regions like in a motor task here. We can look at the whole brain, and we can look at the connections for the whole brain. And admittedly, we have to go back to task-based functional MR in order to say anything about the function of these regions. But we can, with connectivity, look at how the brain is organized functionally. By the way, I'm open to, I don't know how you usually do this, but I'm open to interruptions at any point, and we can argue points or whatever.
Usually interrupt a lot.
Good. Thanks, Bruce. Connectivity provides a potentially more basic measure on how the brain is wired and how different regions are connected to one another. And it provides a whole brain assessment of the functional circuits. So when we talk about connectivity here, we're looking at these very low frequency fluctuations in the brain where we take a time course from one region, let's say this red time course, and we can correlate it with the time course from another region. And these are just resting state fluctuations in the bold signal, the blood oxygenation level signal. And we can look at those correlations throughout the brain, either between specific regions of interest that we're interested in, or between individual voxels, or between a region of interest and the whole brain. So as I said before, these are very low frequency fluctuations. I think there's some newer data showing that there's evidence for some of these signals at higher frequencies as well, but to date most of the work has been at less than 0.1 hertz frequencies. And this is thought to represent brain regions communicating with each other. So it's not directly task related, although you can tweak the connectivity with tasks. And it's not specifically related to consciousness. There was some interest in whether it was an indicator of consciousness. Actually, for those people at the back, there are 3 or 4 or 5 seats up front here if you want to come in. Come on, Van, you can sit at the front here. And there's seats up along the side here as well. But it's possibly circuit tuning. One of my thoughts on this is that the brain is kind of a competitive environment, and if you look at the motor cortex, for example, and you cut off a limb, the adjacent cortical regions will take over that motor cortex pretty rapidly. And so I think you need to maintain these circuits continuously in order to have them. And so that may be responsible for some of the high baseline energy and for some of these low frequency fluctuations. It's been difficult to validate, and in fact this has been around since 1995, and I think the first 10 years of this field, it was sort of, the first 10 years was spent trying to prove that in fact it's not just physiologic noise. If you're looking at correlations, you know, the first demonstration by Biswell was in the I think I have it coming up, but the first demonstration was by Biswell, and basically the carotid arteries feed these symmetric regions of the brain, and so you could have pulsatile artifacts aliased at lower frequencies that could contribute to this. And so a lot of work in the first 10 years went into trying to sort out the physiologic artifacts. And I think since about 2005 on, there's been a really explosive growth in terms of connectivity mapping, and I think fewer people are, or almost everybody thinks that there are some meaningful signals in these correlations. So it's typically obtained, I already mentioned, in the resting state, although it can be obtained during a task. And there is lots of evidence that connectivity can be modified by task or by brain state. But it's basically an intrinsic measure in the sense that work by ourselves, putting healthy normal volunteers under anesthetic, as well as the RACO group at WashU, have shown in monkeys under deep anesthetic that they still find these connections even under deep anesthesia. So it's not specifically related to consciousness, but these are sort of intrinsic correlations which are there. And we could discuss what the task or what the conditions are at rest. And I'm not all that interested in this. There are a number of people that are. But suffice to say, you can tweak the connectivity based on the state or the task that the person is in. So how do we do such an experiment? It's very similar to the what I just showed about the task-based fMRI. We run continuously. We collect these brain volumes. We can realign them. And then we're just looking at different brain regions. So if each of these represents a brain volume, we're going to correlate one region of the brain with the rest of the brain or with other regions. And a high correlation implies strong connectivity, or a low correlation implies limited functional connectivity. So a very simple basis for this method. Here's the first paper, was by Barad Biswell in 1995. And he basically showed that he could get motor cortex activated here with a finger tapping task, and that if he took a seed region in the motor cortex in one side, he basically would light up in a correlation analysis the motor cortex on the other side. And so it was a way to extract kind of the functional organization of the hand motor area by looking at just this resting state data. And I mentioned already the work by the RACO group and Vincent at WashU. And here in a Nature paper in 2007, they showed that monkeys, even under heavy anesthetic, still had connectivity to their front. They could do a seed in the frontal eye fields and still pull up the visual cortex and see strong connectivity. And they did some validation experiments where they did an activation task, they did a tracer task, and they did this connectivity, or not a tracer task, a tracer injection here, and they get the same sort of circuitry of the frontal eye fields to the visual cortex coming up in all three different approaches, sort of validating that these are meaningful connections that they're revealing. And again, then they went on to talk about the default mode area and showing that some of the strongest connections are in the default mode area, and that the default mode area in the monkey is actually very similar to the default mode area in the humans, and that's what this panel shows here. So besides that work showing that it seems to be meaningful, other people and ourselves have shown that these measures of connectivity can be correlated with subject variables. We've just submitted a paper on looking at age effects over the lifespan, and there was a paper recently published looking at kind of development through from youth through the teenage years. And there have been a number of studies that have shown a link between functional connectivity and performance on a specific task. Michelle Hampson has kind of spearheaded this work at Yale, and she's demonstrated reading ability changes or is somewhat predictive of the connectivity between BA39 and Broca's area on the left was somewhat predictive of reading ability. So the stronger you had the connections between those two areas, the better reading ability the person had. And similarly, Michelle has also published work looking at working memory performance and the ability to engage or disengage the default mode area in a working memory task. So most of if we talk about the methodology then, to date, most of the approaches have relied on ROI seed-based studies, including the work of Hampson and Biswell and all these other studies so far. And we can do this in two ways. We can take a seed region, a region that we're interested in, and we can correlate that with every voxel on the whole brain. Or we can do kind of seed to seed, so we can come up with a map of seed regions and correlate that. So basically, you choose a seed region. You say, okay, this is from an activation experiment. Let's say we'll choose this seed region here in the left temporal cortex, and then ask basically this question of what other areas share a similar time course in the resting state to that seed region. And the assumption is that such areas are communicating, and hence these are connected. And this potentially identifies normal functional circuits, so we could do this in a group of normal healthy volunteers, and then you could do this in some patient population, and we could compare those and look for dysfunctional circuits, for example. The question is, how do we define these seed regions? We've used a number of different approaches, as have other people. We have used a combination of kind of anatomic atlases, functional regions, and again, the functional regions are limited by the number of tasks you can do that identify functional regions, and or some combination of these, and I'll talk about each of these a little bit. We've used the Brodmann areas. They're defined online, and you can access those. These are based on cytoarchitecture, so that seems to make sense, but some of the Brodmann areas are really, really large, and you're clearly mixing time courses from different functional subunits within the Brodmann areas, and so that can give you erroneous results. And also anatomic atlases, well, easily defined. Let's say for the hippocampus, you can clearly draw the hippocampus, and that's fairly easy to do. It's much more difficult to do when you get into the cortical frontal lobes, let's say, or the posterior parietal cortex, where the anatomic boundaries are not all that clearly defined. In a basic seed to whole brain connectivity study, we would do something like this. This is a left frontal area defined in a reading task, and you can correlate that then with the rest of the brain, and it shows that you bring out the reading or the language areas, particularly on the left, but you actually bring out the homologs on the right hemisphere as well, and so you can reveal this circuitry. And then once you get a map like this, you could compare this for two different populations, for example. But the key there is how do you define this? And in this case, this was defined with a functional region. Using functional task-based fMRI to define this doesn't generally work for the whole brain because you don't have task data on the whole brain. And so furthermore, the definition, one of the things you have to be aware of is that the definition of the seed may not be appropriate in the two different populations. So that can be a challenge as well. You can come up with a whole combination of these seed regions, and here we have a list of about 30 different regions, and put them in a matrix form like this. So the same seed regions are on this axis as this axis, and then we can correlate them all with each other. And so on the diagonal, it's each seed region correlated with itself. And here, I can't read this, and you probably can't either, but you don't have to. This is seed A. Correlated with B, C, D, E, F, G, etc. So as you go along a row, it's that seed region correlated with everything else. And you can get these kind of connectivity matrices where you can look for patterns, or you can assess network properties of these connectivity matrices, or you can even contrast these connectivity matrices between different groups or between a patient and a control group and things like that. So this approach can be very useful, but it's sensitive to the choice of seed. I have some examples here. I'm going to zoom in on this in a second, just showing the sensitivity to the choice of the ROI. So if we choose an ROI based on this red region here, or the red and the green region, which kind of came up in a task we were doing, if you choose those, this is looking at the red region ROI in this row, and this is looking at the red plus green ROI in this row. And I'll just point out that there's some very different changes in the connectivity pattern. You see a number of regions remain constant, but some regions change significantly. And this is looking higher up in the brain there. Again, here, a frontal region, which is positive there and negative here. And this very strong region in the left cortex here is almost gone in that case. So we can get some substantial differences depending on how we choose those seed regions. And so the main message here is that we're sensitive to these. So seed regions, just to summarize, can be defined anatomically. We can use an atlas such as the Brodmann atlas. We can arbitrarily divide the cortex into subunits, and I think that's been done here actually. Say people have divided the cortex into 1,000 subunits. We can define spheres. People have used spheres of different radii throughout the brain. And as I said, on a limited basis, you can define them functionally. Or one of the projects we've been doing, and I know some work has been done here as well, you can define these functional subunits using the connectivity data itself. So you can use the connectivity data to try to parcellate the cortex and find regions that have similar time courses. And so you can kind of grow a region around a time course and say, let's include everything that has a similar time course, and that'll be one functional subunit. And so we actually have a project at Yale where we're working on this problem, trying to parcellate the cortex into these sort of minimal functional subunits at the resolution of standard group fMRI statistics. Obviously, this, how we define these minimal functional subunits is going to take resolution into consideration, because we know from very high resolution studies we can get down to cortical columns and things like that, and that's not what we're talking about here. So there's open questions in terms of how many subunits, what are the trade-offs in terms of reproducibility and number of subunits. As you go up and try to define the cortex on a finer and finer scale, the reproducibility of some of the data gets worse and worse. And then also, how consistent are these subunits? How much reorganization of each of these subunits occurs as a function of brain state or task or disease? And so this is active work right now that that's underway at our place, and I know it's actually underway here as well.
In general, how symmetric are those correlation matrices, depending on how you define them?
Highly symmetric. Yeah, they're surprisingly highly symmetric. That's what?
Comforting on some level to show you that it actually is.
Yeah. Oh, some could argue it's not comforting, but like for example, with the language cortex, you'd think there'd be more asymmetry in the language cortex, since we're also left dominant, but yet the connectivity data is very, very symmetric in terms of left and the right homologs, and that's a little bit surprising, I think. So an interim summary, what we have a task-free approach here to examining functional connectivity. So we've moved away from task-based fMRI. And we have connectivity between regions that appears to be predictive of performance. The current limitations of resting state connectivity are this need to define seed regions to use for these correlations, and the sensitivity of this approach to the choice of the seed regions. So now we're going to move to kind of a voxel-based approach, this intrinsic connectivity mapping, that looks at doesn't need to define seed regions ahead of time, just looks at the connectivity on a voxel basis, and therefore gets around this seed definition problem. One of the first papers was by Randy Buckner on this, looking at this intrinsic functional connectivity. He mapped, on a voxel level, he mapped the major cortical hubs in the brain, and then he did an analysis of the network properties of those cortical hubs. But he used kind of the intrinsic connectivity measure that I'm going to talk about on a voxel basis in order to identify these hubs, and that was back in 2009. So the contrast mechanism, this is potentially a novel clinical contrast mechanism for looking at the functional organization of the brain. So now, just as in the ROI-based or seed-based approach, you can calculate the connectivity between a single voxel and all the other voxels in the brain. But rather than making a huge matrix for that given voxel, what you can do is you come up with a summary statistic, which in most cases is the measured degree, which is the sum of all the connections to a given voxel. So one of the things is here in doing this, you're going to define some threshold of correlation. Now let's say it's a correlation threshold of 0.25, and you say anything that has a correlation above that is going to be considered a connection, and let's just count the number of connections. And that's what the degree measure is. So this generates an image volume where the intensity of each voxel reflects how strongly that voxel is connected to the rest of the brain. How strongly in the sense of how many connections it has, the number of connections it has. Such maps could be examined directly, but we don't actually know what a normal map should look like. And instead, we collect normative data on a group of healthy volunteers, And then we can look at individual patients relative to that group of volunteers, or we can look at a patient population relative to that group. And now we're operating on the voxel level. This is what a connectivity map looks like. This is for 42 subjects. So the bright areas in the brain here should have more connections, so they're connected to more voxels than the rest of the brain, and the dark areas are connected to fewer voxels. So we can get a grayscale sort of contrast based on this connectivity, and this is the network measure of degree that we're doing here. So this has potential clinical applications in a range of diseases because it's a general approach to assessing functional organization. So we can look at Parkinson's, Alzheimer's, stroke, etc. I'm going to show you a few examples in epilepsy. Yeah.
Todd, that previous image showed some areas that looked like little patches of, different connectivity. I don't know, parietal areas. Is that real, or did you ever, try to back out the anatomic identity of areas that might have different connectivity?
Well, so you can threshold. I'm not sure I get your question, but just that there are bright areas and dark areas. Is that what you're?
Yeah, pretty much. And do those correspond to?
Yeah. So the brightest areas tend to be the default mode network. It turns out, and then I can't think of what the darker areas are kind of the other areas that aren't the default mode network, but parsing it out any more than that, we haven't done that yet.
Are they very sensitive to kind of the threshold you set for what's
Yes, and in fact, that's the whole last part of my talk, is to talk about the threshold. Thank you very much. So here, so I was saying that we could apply this in a number of different diseases, and in fact, I'm going to show you some examples here in epilepsy. So there's a couple of different ways you could do this. For example, we could calculate it for the whole brain. So we take a single voxel and we say, what's it connected to in the whole brain? Or we could do just an ipsilateral connectivity measure where we say, what's it connected to within this hemisphere? Or we could say, what's it connected to in the other hemisphere? So we get an ipsilateral measure, and we can get this for every voxel in the whole brain. And we can get a contralateral measure for every voxel in the whole brain. And we could even compare these and see what proportion of connections are ipsi versus contralateral and things like that. And what I'm going to focus on here for the, basically we're measuring the degree, and this is the sum of all connections greater than a threshold of 0.25, and this is an arbitrary threshold, which I'll discuss. For the epilepsy work, because we're trying to quantify the hemisphere that might have abnormal connectivity, we used an ipsilateral measure with the thought that that would definitely lateralize the hemisphere. If we found an abnormality here, for example, in the contralateral measure, we don't know whether it's due to this tissue right here or if it's due to the connections that this tissue has. And similarly, in the whole brain, you don't know what the source is. So we thought this might be a little easier to interpret. But here's an example of a single patient versus 20 control subjects in those three different measures. And you can see that for, like here on the right medial temporal lobe, you can see, for example, pretty similar results for the ipsi and the bilateral, but not so much for the contralateral. So there are some differences in these measures, and we haven't explored these differences fully yet. So I'm not sure what all these differences mean. But there are cases, so for example, there's some theories in bipolar disorder that it's a disconnection across the hemispheres, and so this difference maybe in ipsi contralateral might be very useful for that sort of assessment.
And what does it map of a single subject versus 20 subjects look like?
This is...
a single non-patient subject, control subject versus 20 control subjects.
Oh, yeah, so we've done that, and that's how we try to establish our thresholds by just pulling one of the normal controls out and finding the threshold so that we have less than 5% spontaneous. And one of the problems that we have in this is sensitivity. So when we do compare a single subject to a group, 20, or we actually have 100 controls now, oftentimes we can't satisfy, we can't correct for multiple comparisons and still have a significant difference, and I'm going to talk about sensitivity as well coming up. So here we get some left medial temporal. This was a patient that supposedly had left medial temporal lobe epilepsy, and certainly we see differences here. We see decreased connectivity here, but we see it kind of bilaterally as well. And one of the things to note is that when you do a seed-based connectivity study between, say, the left hippocampus and the whole brain, the thing that the left hippocampus is most strongly connected to is the right hippocampus. So it's not surprising that in epilepsy, for example, that these seizures that originate in the hippocampus lateralize kind of almost immediately.
So how can you take into account the anatomical differences that would be between the MLT patient and the health patient?
Yeah, well, so here we're not. So these are non-lesional. There's going to be atrophy differences here that could be accounting for some of these problems, but we haven't taken into account anatomic differences at all. So the assumption here in doing a voxel-based comparison is that you're comparing the same anatomy. That's a good point. Yep.
What about, have you looked at the distribution of RNA?
Yep. Coming up.
Excellent.
And so we can compare this with cortical mapping. So a lot of these patients get electrodes implanted. And it turns out in this case, this pink strip here was where the seizures were originating from. So that looks great. But you note that we have a lot of other stuff here. And so one of the things we're doing with this work now is trying to look at network properties. Let's say this becomes a seed region. We threshold this higher. This becomes a seed. We look at how these are connected, and is there something about the network that tells us whether surgery will be successful and these people are not. And so we can register all this data using software developed at Yale and try to sort this out, basically. One of the problems I've just mentioned is that we get all sorts of different areas activated, and we have to somehow narrow that down. So here's a case where it was supposedly right insula, was where the seizures were coming from, and we certainly get that, but we get a lot of other areas active as well. So one of the things to do is that if we get this right insula area and clinical data indicates that it's coming from that area, we could use this as a seed, see which of these other abnormal regions is connected to that right insula, and then we could kind of do a combination of, we want areas that have abnormal intrinsic connectivity and are connected to this suspicious area. And so that's what we did here. Here's a connectivity map where we had a seed in the right insula, or a seed, I think it was right in here. And then we looked at what's connected to that, and you can see bilateral hippocampi are connected, and we can do an intersection, and we can limit then our regions that are highlighted to a little more circumscribed space. But you can see that it's often still bilateral, which may actually account for the poor success rate of many of these surgical interventions. So, but what we've demonstrated here is that we can come up with regions that have altered intrinsic connectivity and are potentially part of the same network. And then we can start to do network analyses on these. And so this is the final composite for that patient. Again, showing this right sort of temporal region. The electrodes also showed that region here. And so this looked like a great example of how our technique really works. And this patient went to surgery, and this is the region that was taken out. This is kind of our region overlaid on the void under there. And the patient still seizes after surgery, so not a great example. But we don't know if that's because, maybe these other regions, which are part of this network, are really responsible. And this was involved. It wasn't the critical node in the network. And so that could potentially explain, and we need a much bigger patient database in order to sort this out. And so we're working on that. These are some examples, other examples where we have the grid and the EEG from the electrocorticography showing seizure onset in L10, and we get a big change in signal there. Again, in some other examples, sorry, that's a little overexposed there. But 3 different examples where we see great results. Stufflebeam from here has published a paper this year in Journal of Neurosurgery where they showed, here's in 5 patients, I think, really good correspondence, basically in the same measures, although here what they did was they had spheres around a region of interest, and they looked at connections in a close proximity to that sphere and also connections outside of a larger sphere. And, sorry, am I talking too loud?
No,
it's echoing a bit, yeah. And we're able to show pretty good correspondence between these, what they found with the grids and what they found with the intrinsic connectivity. So this is promising work, but it's difficult, and we've been doing this for a while and not had this much success. So for an interim summary here, this is potentially an excellent contrast mechanism for assessing functional organization of the brain. We can calculate the connectivity on a single voxel with all other voxels in the brain, summarize this by this degree measure, and generate this volume which reflects the connectivity. There's two problems with this that have already been raised in questions from our astute audience here. That the results can change as a function of the arbitrary threshold, and that the sensitivity appears to be low. So the next steps are then, what can we do to eliminate this arbitrary threshold and to improve the sensitivity? And it turns out that there's one thing that we can do that kind of addresses both of these, and that's, as Bruce asked, can we look at the whole spectrum of connections? As Dr. Rosen asked, what's the sensitivity to threshold? Here's an example showing the sensitivity to threshold. Now remember, a lot of the network theory that's used in this comes from social network theory or disease tracking, where they have binary systems. So think of Facebook. You either have a friend on Facebook or you don't, okay? It's not you don't get some graded level of friendship on Facebook, right? Although you can put them in categories now, yeah. But so that's a binary system, and a lot of the network theory comes from those sorts of systems. In tracking a disease, you either get the disease or you don't. And so those are binary systems. In fMRI, we have this continuous spectrum of connections with correlations from -1 all the way to +1. And it's a continuous spectrum. So we have to make some arbitrary decision somewhere as to what's a connection or not. And as you show here, if we make a connection, if our criteria is low and we say, okay, we only need a correlation of 0.15 in order to determine that these two regions are connected, that's fine. And you can see, as we highlight here, this region, for example, disappears if we go to a higher threshold of 0.45, and it's gone at 0.7, and it's also gone at 0.4. Similarly, this region disappears. And so a lot of these regions, this blue region here, which comes in at a high threshold, isn't here at these lower thresholds. And so as we change our thresholds, we change basically what we see as being connected. By the way, this is a group difference map. So this is comparing low birth weight children versus normal term children at age 18. So they've been following these children for 18 years. And this is comparing their brains now at age 18 based on what their birth weight was. And we can see that the results in that group comparison, and these are decent numbers, almost 20 per group, we can see that these results change with threshold. Yes?
Why do you threshold the network? It's a problem. Why don't you use a weighted network?
We can do that. And in fact, what I'm going to show is that we can look at the whole, well, with a weighted network, you're still making assumptions on weighting, but that is an approach around this. What we do is, I'm sorry, this is not showing up very well either. There's actually the key curves are in the middle here. Can I change the contrast on this? Where's contrast on here? Corrections? Yeah. Thank you. Brightness, contrast. Oh,
Very
great.
bright when we talk.
Yeah, I know. There we go. Aha, it worked. How tough could it be?
You know that, right?
Yeah. Okay, so now you can see these two curves. This is showing the same preterm term groups, and the curves cross here. So in fact, our connectivity in this case can go from negative to nothing to positive, depending on which threshold we use to calculate this degree measure. So we have a real sensitivity here to threshold. And
Why don't you actually just use the curve instead of the...
exactly. So we decided to use the curve. You guys keep jumping ahead. So what we're doing now is we're looking at the whole curve. This is the intrinsic connectivity distribution now. And so we can model this curve. It's essentially like an exponential decay that we always see in MR. So we have an alpha term here, which is the decay rate of the curve, and we have a beta, which really just accounts for a shoulder effect there. And so by looking at the whole curve now, we can get rid of this arbitrary threshold. Alpha, this just shows as you change alpha how the steepness of the curve, the decay rate of the curve changes, and beta is really this shoulder that you can have. And so these are the two terms that characterize this curve. When we put them together, we get these a wide spectrum of shapes that we can fit. And so when we do that, it actually turns out to be quite useful. The sensitivity appears better, and we've eliminated this arbitrary threshold. Now, the one thing that I'll say is that we haven't completely eliminated the threshold, and that we're not looking at the whole curve, because the whole curve goes from -1 to 1. And there are definitely some interesting connections in the negative side of these correlations. But if you wanted to argue, you could say we have an arbitrary threshold now of 0, and we're looking at all the positive connections. But we're considering more of the data, and we're looking at at the entire curve, and we can get away from this arbitrariness here of these different thresholds. And so looking at that, we have this degree measure. This is the initial thing I was talking about where we had a threshold. In this case, it's a threshold of 0.6 for the ICC degree measure. And when we look at our alpha term in the distribution, we get this sort of result here. Now, you can see the results are very similar. This is at a 0.05 corrected threshold. And this, nothing would survive if this was corrected. And so this is at a lower statistical threshold here. But if we lower it, we can see that some of these things are there. It's just these don't survive correction. So we get better sensitivity here, and we've eliminated this arbitrariness of the connectivity threshold. So we think we've examined the sensitivity by looking at...
Justification for the functional form? In other words, you could fit that to a logistic equation.
Yes, no, I don't.
Any number of different equations that will fit that type of...
Yeah, I don't have... And if you have any thoughts on how you might come up with a more meaningful curve-fitting
Never ask Bruce
approach.
I'd
I'd love to know. But yeah, no, this is, so there's an arbitrariness here, and what we chose is to characterize the curves. But yeah, and there's arguments for and against that choice. It's unfortunate that it's a 2-parameter fit, too. I would have much preferred a 1-parameter fit, and that we could fit positives and negatives and have everything. But we didn't do that. So anyway, so we've solved the sensitivity problem, we think, and we've eliminated this arbitrary threshold, or we've at least replaced it with a threshold of 0. And so our results are somewhat more stable and less vulnerable to that threshold.
Have you done any fitting? I mean, have you looked at how well that functional form fits the data?
Yeah, I can't. We have, and in fact, we did try other distributions, and I can't give you any numbers on that though. So I can't tell you how, what the variance was or anything like that. So I presented a whole brain voxel level contrast mechanism based on functional connectivity. This voxel level avoids the need for a priori information on ROI definitions. We examine the entire distribution and avoids this arbitrary threshold problem. It's potentially useful then for comparing patient populations to controls for this sort of genotyping experiments and or phenotyping of various genes and for drug studies. So a number of people at Yale have been instrumental in doing this, and we have software that's freely available that does this. And so I actually do very little of the hands-on stuff myself, so these are the people that actually did all the work. And so thank you very much. I'm happy to answer any questions, and you're welcome to attend this workshop. I'll just put in a little plug for this winter, February, in Whistler, BC. Thank you.